So now we're going to move to our next speaker, Shahragim. I will introduce him. I will let the régisseur come and connect up appropriately as I introduce him. So basically, Shahragim is a specialist on the muscle, and I actually covered his work as well in my lectures to some extent. So of course, the muscle is an organ, unlike the intestine that constantly is regenerating. The muscle doesn't have to regenerate, but definitely does upon injury, etc. So dramatic changes happen, and Shahragim has been one of the pioneers in really thinking about how skeletal muscle stem cells self-renew via symmetric and asymmetric divisions, trying to get a molecular understanding of the basis of this transition or shift to asymmetric divisions in different contexts, and also exploring how the stem cell niche is defined. So without further ado, Shahragim, who is at the Pasteur Institute and has been there for many years and actually is one of the big actors in France as well for the French Stem Cell Society, and I want to thank him for all the good things that he does there too. So Shahragim, thank you so much for coming, and we look forward to hearing you.
Thank you very much, Edith, for this generous introduction and for allowing us to come out of our shells and the metamorphosis for an in-presence meeting. This is very strange, and I'm still adapting. So while we're waiting for the slides to come on, I was asked to discuss some issues regarding memory and epigenetics. And now I can remove my mask. Unfortunately, we have some recent results that address this issue. So I'm going to be focusing, as Edith said, on skeletal muscle, on properties, and in time and in space. So to put things into context, I'd like to remind you that there was a seminal series of experiments that were done in the 80s by Weintraub and colleagues, where a fibroblast was converted into a muscle cell through the action of MyoD or other transcription factors of the same family, MYF5, MR4, myogenin. And this was a key result that was actually the springboard, an intellectual springboard, leading to Yamanaka's discovery many years later. What I'd like to insist on here, although we've forgotten about this type of experiment, there's some details that still need to be ironed out. One of them is a transcriptome, and the epigenome is defined in this fibroblast, and it's fixed to a certain state, and it breathes with a certain range of movement. In the muscle cell, in a true muscle cell, it has its own transcriptome and its own epigenome, and I'm going to address that issue a little bit later. But when we convert this to this, is the transcriptome fully converted and is epigenome fully converted? And the answer is no, not always. So although we get a phenotypic output of a muscle fiber that's contracting in a dish, in some cases, even in vivo, when the cell is fusing into the fiber, there you have a partial reprogramming of that genome, so which is interesting. So these factors are very dominant to push that phenotype, but the underlying structure is not completely worked out. And there's still a lot to do in this field. And there are other types of experiments fusing two cells, a fibroblast and a muscle cell, instead of reprogramming, so the heterokaryon types of experiments. And in those cases, the muscle genes activate the endogenous muscle genes in the fibroblast cell, but that nucleus is not fully reprogrammed, not always. So that's interesting. And a considerable amount of work has been done in this field, as you heard from Hans, fibroblast to iPS transition, which has its own epigenome and its own epigenome. And this is a multi-step process. And in some cases, it's much better studied than this model, given the number of labs that are working on it. And there's still some things to work out in terms of the epigenome, at least, and how this cellular memory is being transmitted. So in that context, I'd like to discuss to what extent does history impact contemporary behavior, as our personalities are defined by those who mentored us, who educated us, and even indoctrinated us. Can we get rid of some of that? I'm not that convinced. And can a cell in our body get rid of that? We can see that to a certain extent, but not in all cases. So in the case of muscle, we're going to use and exploit the diversity that we've seen in muscle heterogeneity to answer those questions and challenge this cell state at the transcriptome and epigenome level. Now, to put you all into context, as many of the internal organs of our body are highly localized, this is not true for skeletal muscle, which is found throughout the body, like bone and like skin. So this poses the obvious question, are these muscles similar or different? And I'm not talking about just fast and slow muscles, the sprinter fast and the marathon runner slow muscles. I'm talking about an underlying diversity that goes beyond just contractility and function, but the underlying transcriptional networks. So we're going to look at the transcriptome epigenome, and does this degree of heterogeneity that is found throughout the body impact on the emergence and the function of the tissue, and in the context of adult regeneration, disease, and aging. So we haven't addressed all of these questions, but we're starting to dabble into that. And just as a reminder again, skeletal muscle is involved in many types of functions, from nutrient reserves to posture, and it's an excellent model for studying all of these conditions, from homeostasis, quiescence to cancer phenotypes and aging. And unfortunately, most of the field, experimental field, that has been interrogating these issues has focused on muscles below the knee. And that's the bulk of our literature. And so one asks the question, well, if a muscle is going to contract, it's attached from bone to bone, and it's just going to contract and allow you to move, it's a voluntary muscle, unlike smooth muscle and cardiac muscle. Big deal, because you're just going to do one thing and you're just going to contract. Well, it turns out that notion is satisfied by this result that came out in the early 90s, one of the first knockouts that were done in the early 90s, where transcription factors, the ones that I mentioned already, MyD, MR4, and MF5, if you knock out all three transcription factors, you eliminate all skeletal muscles in the body. And this is satisfying in saying, okay, all skeletal muscles are alike, and they all need these transcription factors to acquire their fate from a naive upstream cell. And don't forget, there is an upstream cell giving rise to this progenitor that's going to give rise to these myoblasts. So we get rid of all the muscles in the body. But unfortunately, it wasn't as easy and simple as that, because a few years later, we did another series of knockouts. The genes here are not necessary to remember, but just look at the scheme, where we eliminated all body muscles, but the head muscles were intact. And this was a bit of a shock. And one of the first experiments that told us that there's a dramatic difference between the stem cells that are born in the head and the stem cells that are born in the body in terms of their gene regulatory network. And a few years after that, we did another series of combination of mutants where the body muscles were intact and the most of the head, except the extraocular, most of the head muscles were gone. So this told us that really there's an underlying network that was clearly defined and different. And if you were an intelligent designer, you wouldn't do this. I think the intelligent designer went out for coffee when this was being made, because you would try to keep things simple and have the same genetic regulatory network. And perhaps it wasn't the intelligent designer, and perhaps it is scars left from evolution, because as a very quick reminder, all the muscles in the body are arising from these segmented structures called somites from the neck down. But from the neck up, the same paraxial mesoderm in line with the somites is unsegmented and has distinct evolutionary origins if we go back to our earliest chordates. So there's an underlying evolutionary scar that might be explaining some of these distinct gene regulatory networks in the upstream population before cells acquire their identity by funneling through these key myogenic regulators. So we're modular in the embryo, and is this modularity kept as a memory? Is this just a remnant in development and everything equalizes in the adult? And what is the impact on the stem cell and the fiber properties in the adult in that case? Now, I'd like to remind you what's happening in the clinic. In the clinic, there's some devastating myopathic diseases where some muscles are affected and others are not. This has been documented for decades and decades, and it still remains one of the big mysteries in the field. If all skeletal muscles are alike and they just contract, why do you have, in this severe case of Duchenne muscular dystrophy, almost all muscles are attacked, and when these individuals die, their extraoculars are still functioning in their head? So we start to use extraocular muscle and cranial muscles. and explore them and ask, well, how are they different from the limb? And we do know that the extraocular muscles, when you put them in culture, they have a higher proliferative rate, and when you transplant them, they have a higher transplantation capacity. So going from that, we then compared the extraocular muscles and this limb muscle, tibialis anterior, in mice. And the stories that I'll be telling you today was done by 3 very talented individuals, 2 former PhD students who continued to finish their work in the lab, Maria and Alex, and a very talented senior scientist, CNRS scientist, Glenda, who's in the lab. And Alex established the RNA-seq pipeline, which I'm going to tell you about very briefly. So as a reminder, these somites establish muscles in the body, but what's happening in the head is this unsegmented paraxial mesoderm that I told you about. That's going to give rise to the pharyngeal arches. Now, we have about 5 pharyngeal arches. Our ancestors can have up to 15 pharyngeal arches, and these give rise to all our craniofacial muscles, esophagus and laryngeal muscles. And the extraocular muscles are sitting up there outside the arches and just minimally related to them. So when we looked at these and looked at the transcription factors that program these, we know that PAX3 is doing the body, TBX1 largely the head, but the extraocular muscles escape TBX1, this homeobox transcription factor, and it's exclusively initiated by PITX2. And then, of course, we zoom in and require identity through the myogenic regulatory factors, and this, through development, gives rise to fibers and future muscle stem cells or satellite cells at later stages. So what we did, what Alex did, is to establish distinct single-cell RNA-seq profiles of craniofacial development and ask to what extent, even within the cranium, is there heterogeneity. And I'm not going to go into detail, but there are different types of genetic tools. A generic tool which identifies all the mesoderm that's coming off the head. A more precise tool, which is MIF5, the first initiator of myogenesis. And both of these are Cree-driven onto a ROSA MTMG, that means permanent marking of those cells. And then contemporary label with MIF5 GFP at later stages, with dissections of those areas that you see indicated over here. And to summarize, a lot of data, I'm not going to inflict the pipelines on you. We see, if you look at this embryo and the craniofacial, and we're looking face on over here, the extraoculars and the arches, we see distinct word clouds that are coming up even within the cranial muscle development. And there are key transcription factors. And in fact, if you look at the pathways, there are key transcription factors and growth regulators that are specific even within subsets of arches. And we know the first couple of arches are giving craniofacial muscles, and the caudal arches are giving laryngeal and esophagus muscles. So here, this is surprising to us because there are several transcription factors that are unique to all of these subsets, and beyond just the TBX1 PITX2 that I was telling you about, and upstream of acquisition of identity. So then we decided to look a little bit more in detail, and Alex used this approach by exploiting transcription factor modules. I'm not going to go into detail, I'll just put that reference over there. And basically what he did was look at all targets of transcription factors and then look at the transcription factors that were targets of those in a hierarchical fashion, and more downstream, and more downstream, and eventually get rid of the targets and look at the flow of transcription factors by looking at the binding sites of the sequences that they bind. And so to isolate this transcriptional factor network, it took quite a bit of effort on Alex's part, but he managed to do that with all the craniofacial muscles as they were emerging in the embryo. And this is what we get, a fuzzball, and I'm not going to bother you by this, but I would like to highlight that there are modules of differentiation that are segregating out, modules for the extraocular muscles that are independent, and these are all transcription factors. Modules for the second and first pharyngeal arches that are coming out. So having that as a resource, we now ask the question, now what is happening? Is this just a curiosity during development, or is that persisting and defining what's happening in the adult as the adult is modular? So I'm going to address that issue by looking at heterogeneity in the cranial and semitic lineages in the adult now. So if we go to the adult and we isolate directly from the adult mouse using a transgenic PAX7 GFP that marks all muscle stem cells in the adult, and isolate those and take the extraocular as an example and the limb as an example. In blue, you see the extraocular. In dark purple, you see the tibialis anterior, which is the limb. You see immediately that these two populations are segregating out at a single cell RNA-seq level, which is very satisfying and unusual. And if you look at MIF5, which is the first, which is an upstream transcription factor, it's relatively homogeneously expressed in both populations, but MyoD, which is a clear marker of cells that are activating, I remind you, these cells are quiescent in the adult. When they're activating, the first marker at the transcript and the protein level that is a reliable indicator of activation is MyoD. And MyoD is much more active at the transcript level, much more active in the limb than in its extraocular, telling us that the extraocular are holding back in an upstream identity, whereas a limb is moving forward, so to speak. And if we take these cells now, put them in culture, and this is interesting because they've been cultured for 4 days, so they've undergone at least 4 or 5 cell divisions. And you'd think 4 or 5 cell divisions, you'd forget about your niche. You've equalized and you've been cow serum for so many days. Well, it turns out that that's not the case because that memory persists. And the upper branch is extraocular, the lower branch is the limb, and they're very distinct at the single cell level, and the extraoculars are continuing to proliferate abundantly. Then they start to converge at the differentiation step. So we do have a memory that's distinct from the embryo to the adult, and we do have persistence of that distinction in the adult, and we have a persistence of that distinction after 4 days in culture. They haven't shed that memory of their niche. The intrinsic properties are still quite dominant. So that's the conclusion on that part. And if we look at upstream markers like PAX7, PAX7 is dominantly expressed in the extraocular muscles, and the differentiation marker myogenin is dominantly expressed in the limb muscles. So what can we do to challenge that phenotype now? And I'm going to explain some work that was done by a very, very talented postdoc in the lab. We just got a CNRS position, Brendan Ivano. And this was a very nice collaboration that we did over several years with the Wolf Reich lab, who provided the high technology of doing simultaneous methylome and transcriptome on cells. So in this case, we know that the extraocular and the limb muscles are very distinct, the stem cell populations. And what Brendan did is isolate those populations, went to the fax machine, and interrogated those cells simultaneously looking at the transcriptome and the methylome with Wolf-Reich's infrastructure. And if we look at the transcriptome, we see, as I showed you earlier, on a PCA plot, the transcriptome of the extraocular and the limb is distinct. I showed you that earlier as well. And there are Hox genes that are coming up in the limb. We do know that they're there, and they are not there in the extraocular. So that's very satisfying, and I'm going to come back to that point. And we can validate them by RT-PCR, and that's the take-home message that I gave you right there. So what about the Hox code? Well, the Hox code is flat. The Hox genes are not expressed in the head and not in the muscle stem cells in the head. Yet they are expressed, at least HOX-A and HOX-C locus are expressed in the limb. And what about the methylation profile? Well, the methylation of the promoters was surprising to us, and I'll mention why later, was not very different between the extraocular and the limb. Yet the methylation at the enhancers was very distinct. And don't forget, we're sampling the genome. We're not doing a whole genome analysis. We're sampling the genome here because of the depth. So then what Brendan decided to do was challenge the system and say, okay, I'm going to transplant the extraocular muscles into the limb and ask, are you going to keep your memory or are you going to adapt to the heterotopic location? So first round, RNA-seq and bisulfite simultaneous sequencing with one aliquot, and another aliquot was taken and transplanted. Now, one very important control that we did, I'm glad that we did it, is to take limb isolated cells and transplant them back into the limb, and compare that to extraocular isolated cells into the limb, and wait 3 to 4 weeks after regeneration is complete, isolate the cells, and examine. Well, it turns out that the pre-graft extraocular muscles in light blue are sitting very far apart from the post-graft extraocular muscles in dark blue, which have been adjusted for the transplantation. And I can tell you, just the simple act of transplantation has modified the transcriptome, and we had to normalize against that. And if we didn't do that, we wouldn't have seen this effect. And so we can validate this by RT-PCR. That's fine. What about the Hox code? Well, I told you that the Hox code was flat. There are no Hox genes expressed in the extraocular muscle stem cells. What happens when you put them into the limb? Well, this is very satisfying because they activate the HOX-A and HOX-C cluster as the limb ones do. So they are adapting. We say, well, this is fantastic because the cells completely adapt to their endogenous environment. Well, that's part of the story. Because if we look in more detail at the full transcriptome, we find that 10% of the genes that were in the extraocular resist that transplantation. They do not adopt their limb phenotype. And these are, a lot of these are transcription factors, signaling pathways, etc. The bulk of the genes, over 50%, are fully responsive, and there's a class of intermediate genes that are adapting. Now, there are subtleties in this experiment I'd like to think about, and that's the lack of control and the total lack of control that we have in this kind of experiment. Because we transplant the cells, but we have no idea how many cell divisions each cell does during that moment. And so you would speculate that the number of mitoses that the cell does might encourage it to adapt a more heterotopic fate. And we have no indication of how many mitoses these cells do. We just take the cells out and do an average. So I'd like you to keep that in mind. So that's a transcriptome. What about the methylome? Well, as I told you earlier, the promoters are not showing any distinct signatures at the promoter level for methylation profile. But as I showed you earlier, the enhancers do. So the enhancers of genes that are adapting their transcriptome are being modified and adapting the methylation profile of the endogenous host. And those are the take-home messages there. Now you'd say, well, okay, cellular memory, you can reprogram to a certain extent, grosso modo, and there's some resistant genes. That's fine, okay? But what about doing transplantation the other way? Well, I highlighted a paper that just came out from our colleagues, Yusuke Ono in Japan, where they took limb cells and transplanted them into the masseter. Now, they did the experiment slightly differently because they irradiated the masseter. And when you irradiate the masseter, you kill all endogenous cells. Whereas in our case, we weren't irradiating, and the transplanted cells were competing with the endogenous cells. That's the first point. Second point is that they sampled at 14 days. 14 days of regeneration is not complete. All right? And we waited 4 weeks for regeneration to be sort of complete. And in their case, they find that they have higher levels of HOX-A10, HOX-A10 being the limb, in the limb, in the in the limb cells that are engrafted in the limb, but the masseter is not responding the same way. So the HOX gene in their case was not being reset, and in our case, it was being reset. So now we have an experimental system between their lab and our lab that we can start to interrogate and ask, is it a difference between extraocular and masseter, or is it an experimental paradigm that is different? All right, and so I summarize this part on the right side, asking how regional identity and specificity is established. I showed you some data in the embryo, and we can challenge those regional specificities by transplantation. And looking at the transcriptome and methylome, what are the molecular mechanisms? And what we have done, which I have not discussed today, is also looked at what happens to young stem cells and old stem cells at the transcriptome methylome level. And this was one of the first studies that came out looking at young and old at a single cell transcriptome methylome. And the take-home message of that was that as the animal is getting older, its transcriptome is getting more and more chaotic, and its methylation of promoters in this case was getting chaotic as well. Whereas in this case, it's the enhancers that were being modulated. So there is no universal rule. It depends on the experimental setup that you're looking at, whether you're looking at in a context of regular aging or you're looking at a context of transplantations. And I'd like to, I think I have just a couple of minutes left. I won't talk about distinct states of quiescence. I'd like to remind you, this is, I haven't shown this photo before here yet. This is a muscle stem cell, and it's very particular, unlike almost every other stem cell in the body, in the sense that it is not in direct contact with its niche at all. So from birth, around birth, or a couple of days before birth, this stem cell becomes ensheathed in the basement membrane that prohibits direct contact with niche cells that are outside, whether it's vasculature, fibroblasts, etc. And the only intimate contact it has is with the muscle fiber. And there are multiple pathways that are there keeping this cell quiescent. And of course, they activate when you damage the muscle. So I'd like to tell in 2 slides one story which actually addresses stem cells and what they do in almost every tissue, those that are quiescent. And this is the work that was done by a very talented PhD student in the lab, Mariam Baghdadi, who followed up from work that was done with David Castell, who did some really nice small RNA sequencing to identify microRNAs. And basically, this is the pathway that we identified, that Notch is activating a mirror, microRNA 708, that keeps cells from migrating. So they're parked in their niche, and they're not going to go when they're quiescent. And you have to get rid of this microRNA to start migrating and to start doing something during regeneration. So given this model, we asked the more universal question: when cells become quiescent, when neuronal stem cells, hematopoietic stem cells, muscle stem cells become quiescent, they do not do that in one fell swoop. They were proliferating at some time historically. Then they become quiescent by increasing their notch levels. Now we know that. They're going from low to high. And they get out of quiescence by decreasing their notch levels. In fact, if you persist with notch, you don't get out of quiescence. So you have to get rid of notch to get out of quiescence. And that raised one issue. And the issue is every stem cell that is going to go under quiescence, that is mesenchymal and motile, not epithelial, but mesenchymal, has to negotiate 2 decisions. When do I do my last mitosis, and when do I stop migrating? Now, do they stop migrating, then divide and sit? Or do they divide, continue to migrate, then sit? And we would propose that microRNA is involved in this step. The cell mitosis continues to migrate, microRNA comes up, parks that cell, and the cell becomes quiescent. And the reason why we say that is not by direct evidence, but indirect evidence. If we look at light sheet scanning of the whole muscle and look at the disposition of all the muscle stem cells, our lab and hundreds of labs have failed to produce an image of 2 quiescent stem cells sitting side by side. I'm really begging for an image like that, and we don't see one. In fact, they're scattered like this, and it's a very sad story. It's a lonely stem cell all by itself. And this is like the hematopoietic stem cell all by itself, unlike the neuronal stem cell, which is sitting in a cluster. So the hematopoietic stem cell would probably be undergoing this kind of decision as well. Mitose, separate from my sister, then park, and be surrounded by a niche. And so there are other models, and we don't think they're as probable, and we would suggest that the last mitosis is negotiated first, then the migration step. So I'd like to end by summarizing what I talked about. I highlighted muscle diversity and heterogeneity from the embryo to the adult. Now we have a great number of tools to look at all the modularity that exists in the head and in the body. And we have gene regulatory networks and transcription factors and signaling pathways that are very distinct. And this is very surprising. If muscle in the end is going to be contractile apparatus, why put so much effort making the upstream population so distinct? I highlighted some heterotropic transplantation experiments where we can challenge this plasticity and ask, are you fully going to reprogram or not? Can you be changed, and to what extent? And I mentioned some words about the stem cell and its niche. And in the end, the most important slide, I thank the people along the way who did the work, Brendan, Maria, Alex, and Glenda, and a very nice collaboration over the years with Wolf Reich's lab, who really provided the infrastructure for the bisulfite and RNA sequencing. And of course, our sponsors and you for your attention. Be happy to take any questions.